Good evening, everyone. Test. Good evening, everyone. My name is Milot Mirdita. I'm from the Steininger Lab at the Seoul National University in Korea. Today, I'm excited to present you my work on Petasearch, which is a new, still work in progress, but a very sensitive tool to do scalable searches against billions of proteins. Some of you might know me already from some of our big tools that I contribute to. Among these are FoldSeq, MMSeq, and ClubFold. The first two are aligners, specifically for structures and for sequences. And the later one is a tool to predict protein structures easily in your web browser. So in today's talk, I want to talk about biological sequence search in a somewhat different way from our previous talk. Here we want to search for proteins, and specifically we want to find homologs. And for homolog detection is a key tool we have for answering various questions in biology, from finding pathways, phylogenies, structures, taxonomies, and many, many more applications. The big metagenomic databases that we have today, protein metagenomic databases that we have today now contain tens of billions of proteins, and they are the treasure troves for information. For example, Ovchinnikov et al. Have shown already in 2017 that using metagenomic proteins, they can much increase the number of proteins that they can successfully predict. And in recent times, AlphaFold 2 has integrated metagenomic protein databases, the BFD and Magnify specifically, as their source to build high quality and highly diverse MSAs for structure prediction. These proteins generally are extracted in various ways from the experiments stored within the SRA, the sequencing read archive. The SRA is a vast repository of sequencing experiments. And in the slide that I got from Artem, and the next two slides that I thankfully got from him, he describes SRA as a library of Alexandria for genetics. The SRA has massively grown in recent times. For example, between 2018 and 2020, they added 10 million samples. To date, it contains over 40 petabases of publicly accessible data. And there were various efforts to make this data searchable. One of these is Ceratus. And Ceratus, I won't go into it much, but Ceratus is a cloud-based architecture to be able to scale to the database searches to the size of SRA. And what they focused here on to have very low costs per sample. And they were one of the, I guess, first groups to show that it's possible to search for petabases efficiently. Here in this instance, they have shown that they were able to search through 1 million of NGS samples in a day for a cost of ½ cent per sample, which is quite impressive. We have recently used the Ceratus search to search through 22 petabytes of SRA data. And we did this to improve ClubFold predictions, so protein structure predictions. You can find details in our preprint, petascale homology search for structure prediction, and later in the poster session at B039. Briefly, what we did here is we took all CASP15 proteins that were up for prediction. And after CASP concluded, we took them all in one batch search and put them into Ceratus and searched against these 22 petabytes of data and combined them with default ClubFold MSAs. Doing this, we were able to much increase our performance. We would have ranked, if we had this procedure available during CASP, we would have ranked in 3rd place instead of 11th place. Please visit the poster for more details for this. However, the issue here is that even at ½ cent per library, this becomes very quickly expensive when you search for millions of data sets. So our goal is to build a new algorithm that can search through tens of billions or hundreds of billions of proteins in still a sensitive manner on a single server and has results within minutes. So ultimately, my goal is to integrate this with a ClubFold, so it should be able to handle our daily ClubFold volume of MSAs, which is 20 to 30,000 MSAs per day. Petasearch, I finally get to the actual topic. The key advantages here are that we efficiently store the sequences in k-mers in efficient encodings, and that we are able to process through terabytes of data in minutes, while still reaching relatively high sensitivities of, for example, a query index diamond. And we also are able to use profile searches, query profiles in this case, which further increase our sensitivity. So the key insight here is that new developments hardware happen constantly, and we can algorithmically exploit these. The insight here is that we now have NVMe drives, which offer enormous read bandwidths. For PCIe 3 generation NVMe drives, these offer 4 gigabytes per second, for example, and newer generations even more. So if you stuff a server fully with these drives, you get enormous read bandwidths available suddenly. So a server with 20 NVMe drives would have an aggregate bandwidth of over 80 gigabytes per second. So to be able to exploit this read bandwidth in an algorithm, we first have to pre-process our data. So we take proteins from various metagenomic extracted databases like the IMG or Ceratus or Metaclust, and extract k-mers from these. We do this in an efficient encoding that only takes on average 2 bytes of space per k-mer, and we also need to store where this k-mer came from. So we spend another 4 bytes to store an identifier. Additionally, in later stages, we need the sequence information. For this, we try to also reduce redundancy in our encoding and only store 5 bit per amino acid, as there's only 20 amino acids. In the very first stage, we extract all the k-mers from user-supplied input query in FASTA format and store the query identifier, reserve space later for a target identifier, store a numeric encoding of the k-mer, and the position of the k-mer inside the query. Now here the crucial part comes that we can very efficiently now and very quickly compare this k-mer table linearly against the preprocessed target databases. And we can do this with high bandwidth, and on a server that is full of NVMEs, we can now use high aggregate read bandwidth that is actually available, so up to 80 gigabytes per second. Next, we apply a few criteria to reduce spurious k-mer matches. We do this to ensure that we have that we run more expensive algorithms for as few matches as possible later. The first step here is to demand at least 2 k-mer matches between a carrier and target, and reject all that didn't make it to this threshold. And then another insight here is to demand k-mer matches that are on closed diagonals within each other. This mirrors a strategy we had in MM6, where we demand double consecutive k-mer matches. In the final step, we use efficient implementation of Swift Waterman, which is called Block Aligner. This is a Rust library which can very quickly, at orders of magnitude faster speed, align things at a slight drop in accuracy compared to traditional Swift Waterman implementations. It does so by using a dynamically banded alignment algorithm. And the final output here is then an MM6 or BLAST compatible result that can be used with any downstream tools, or we can generate MSAs or whatever. To show that Petasearch works well, we measure the sensitivity in a UniProt benchmark. Here we sample 1 million UniProt sequences and cluster them at different sequence identity thresholds. I will go only through a 30% example through this case, but we do repeat this procedure for the different sequence identity thresholds. We annotate the sequences with SCOP. And from the annotated fraction, we take 5% as queries and 95% as targets. For the queries, we reserve, we reverse the non-SCOP positions, and for the targets, we shuffle the non-SCOP positions. This ensures that that only the best SCOP target can find each other, and everything else should not be able to find each other and should be a false positive. So true positives are hits that have a SCOP hit of at least the given clustering threshold of, here in this case, 30%. Here in this result, we can see that Petasearch generally works well up to 60-50% sequence identity, which is not very remote. And then its performance drops off quickly at very remote sequence identity levels down to 30%. However, we do have another trick up the sleeve. If we supply profile, so PSSM input to Petasearch, we can recover our sensitivities and still be sensitive down to 30% sequence identity. However, at slightly increased false positive rates. The main advantage here of our tool is with the strategy we can search in a mere minutes against billions of proteins. Here in this case, we have 10 terabytes of data on the target site, about 9.3 terabytes, and we take 3 megabytes of queries. This data is supposed to fully fill 20 NVMe drives, and we can see that traditional tools like MM6, our tool, and Diamond, a competitor, are still able to deal with this enormous amount of data. However, we don't want anyone to wait 2 days for a result. So we show that Petasearch is able to process this amount of data in about 15 minutes. Okay, so to summarize, Petasearch is a fast and sensitive tool to search for billions of proteins. We have designed it as a single server application that exploits this modern hardware that's now available. The work is still quite a work in progress. However, I hope we have a preprint up in a few weeks. And near term that we will be able to offer this as a web service to the community that they can use in their applications, and also we can use to build better protein predictions within CollabFold. And we have a bunch of stickers. Please visit us at our various posters. My poster session was already yesterday, so sadly, don't get to talk to, well, if you see me, talk to me. Okay. Thanks to all the people who have contributed to this and our very cool lab, and all the institutions that fund us. Thank you.
Excellent. Thank you. Questions for our speaker?
Hi, great talk.
Thank you.
I'm wondering if you have a sense of how much of the speed up is due to hardware versus algorithmic.
I mean, this algorithm was developed to exploit a certain hardware, just like we did for MM6. So people at blast ages didn't have like hundreds of gigabytes or terabytes of RAM. If they had hardware in that time, they would have probably exploited this hardware. So now we have a different kind of hardware that we designed specifically algorithms for. So I would answer that as a combination of both.
Thank you.
That was great. Thank you.
Thanks.
When you incorporate the PSSM for the remote
Yes.
homologs, do you incorporate a PSSM that's already converted into the bit structure that you have, the compressed version, or do you have to then use like standard amino acid PSSM?
It's an MM6 PSSM, so there's a few tricks involved there. It's a bit too much to go into detail.